Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about homopolymer tailing. Homopolymer tailing is a process that helps in the addition of a homopolymer tail to the 3' end of a double-stranded DNA molecule. What is homopolymer? The polymer, which is composed of identical repeating units, is known as homopolymer. Already you know about restriction endonuclease. Restriction endonuclease are the enzyme which recognize and cleave DNA at or near specific recognition site known as restriction site. Restriction endonuclease produce two type of cuts. One is straight cut and the other is staggered cut. What is straight cut? If both the strands of a double-stranded DNA molecule are cleaved at the same position, then we will get two DNA fragments. This is one DNA fragment and this is another DNA fragment. So the ends of this strand of DNA are at the same position. There is no single stranded projection. So such type of ends are called as blunt ends. When the restriction endonuclease makes straight cut, then the ends are blunted. And there are some other restriction endonuclease, particularly type 2 restriction enzyme, which make staggered cuts. What is staggered cuts? Now, staggered cuts means the two strands of a double-stranded DNA molecule will be cleaved at different position. So, by cleaving this DNA molecule at different position, we will also get two DNA molecule. This is one DNA molecule or DNA fragment, and this is another DNA fragment. So in this two DNA fragment, you see there is some single stranded protruding tail. This is the protruding end. Such type of single stranded protruding end is known as sticky end or cohesive end. Ligation of blunt-ended DNA molecule is less efficient. It requires higher concentration of DNA and DNA ligase, whereas ligation of DNA fragments having sticky end or cohesive end is more efficient. Homopolymer tailing is a process that is useful to transform blunt end into cohesive end, by which ligation can easily occur. So let us understand the process clearly. For example, this is a vector and this vector is having blunt ends. Similarly, this is the DNA inset. This DNA inset we want to clone into this vector. This vector is blunt-ended. This DNA inset is also blunt-ended. So ligation of such blunt-ended DNA molecules are less efficient. So we use an enzyme terminal transferase. Terminal transferase is an enzyme that adds numerous nucleotides to the 3' end of double-stranded DNA molecule. The primary substrate for terminal transferase is a 3' overhang. But this enzyme can also add nucleotide to the blunt end or recessed end, but with lesser efficiency. So, this is the vector molecule and this is the DNA inset. We want to clone this DNA inset into this vector, for example, a plasmid. But this vector and this DNA inset are blunt-ended. So, by homopolymer tailing, we will convert blunt end into cohesive end, by which ligation will easily occur. So homopolymer tailing facilitates the ligation of two DNA molecules by creating sticky or cohesive end at the 3' end. So this vector is treated with the enzyme terminal transferase and DGTP. So by incorporating DGTP in the reaction mixture with the help of enzyme terminal transferase, we can add a homopolymer tail that consists of many guanosine monophosphate unit. So this is a homopolymer tail and this is a homopolymer tail. Both of these homopolymer tails are composed of repeating guanosine monophosphate. Similarly, the DNA insert is also treated with terminal transferase and the substrate is DCTP. So by incorporating DCTP into the reaction mixture with the help of enzyme terminal transferase, it is possible to add a homopolymer tail that consists of repeating unit of cytidine monophosphate. And this homopolymer tail is complementary to that homopolymer tail. When we are mixing this vector DNA with the insert DNA, annealing occurs very easily because this homopolymer tail is complementary to this homopolymer tail because guanine is complementary to cytosine. So the presence of complementary cohesive ends at the 3' end bring two DNA molecule into close proximity by which ligation can occur very easily with the help of enzyme DNA ligase. So friends, this is about the process of homopolymer tailing. So by the process of homopolymer tailing, we can also incorporate radio-labeled nucleotides at the end of DNA molecule. So homopolymer tailing is also useful in nucleic acid labeling. Please like and share this video and also subscribe my YouTube channel known as Sam Online Class. Give your comments. See you, bye bye.